Good afternoon, ladies and gentlemen. Welcome to Carestream Molecular Imaging's presentation, Multimodal Imaging of Drug Delivery. Due to the number of people here today, we have muted all attendees. If you have any questions during the program, please type them into the question box to your right. We will try to answer as many questions as possible during this time, and will contact you directly if we're unable to answer your questions online. I am now pleased to introduce you to today's speaker, Dr. Seth Gammon, Global Product Manager for In Vivo Imaging Systems for Carestream Molecular Imaging.
Good morning, everyone. My name is Seth Gammon, and I would like to welcome you to our webinar today. Today I'm going to talk to you about multimodal imaging of drug delivery, and in particular focus on novel drug delivery systems. First, just a quick disclaimer, there's a lot of intellectual property that is out there covering multimodal imaging, and we just like to let people know about that. So when can in vivo imaging help you with evaluating drugs, and in particular in the drug discovery process? Broadly, molecular imaging can help in quite a few different areas. It can help in the discovery phase in that you can look at existing in vivo models of disease and quantify those biological processes at the cellular and tissue level to just better understand the progression and mechanism of these diseases. In addition, a lot of in vivo imaging systems, including our own in vivo extreme and multispectral platforms, allow you to image cells in the imaging system in order to interrogate those fundamental biological processes ex vivo. This is also quite useful in the development phase when you have multiple compounds that you're trying to evaluate the efficacy and dose response of those compounds on different genetic pathways. So you could take transformed cells, put them ex vivo, and then study whether the promoters turn on and turn off by looking at whether the light turns on or turns off, whether you're looking at a luciferase reporter or fluorescent protein. The area that I want to focus on today, though, is how can I evaluate quickly in vivo novel drug delivery methods. And preclinical molecular imaging, in particular optical molecular imaging, allows researchers a really great opportunity to rapidly validate the approximate pharmacokinetics and the specificity of these drug delivery vehicles. Do they get to the target they're intended to reach? How specific are they? How fast can those drugs get there? A question that often comes up is, when should I use optical imaging like near-infrared fluorescence versus when should I use nuclear imaging like PET SPECT and CT, or PET and SPECT, excuse me, for evaluating these novel drug delivery vehicles? And really the answer has mostly to do with the size of your system. So if you're looking at a small molecule, then adding a very large, say 1,000+ molecular weight near-infrared fluorophore is going to significantly affect the distribution of that small molecule. This would not be a good time to utilize an optical probe. Indeed, this has been borne out recently in papers that have shown that glucose labeled with near-infrared fluorophores in fact does not behave well or doesn't behave like glucose. In this case, it's a much better choice to label those small molecules with radioactive isostares, typically PET isotopes. In this case, fluorine being the most common isostare that's out there used, although certainly you can replace carbons using carbon-11. When you get up to the size of your delivery agent being a targeting agent, being a peptide, a nanoparticle, or a micelle, now you have the opportunity to utilize either SPECT probes or optical probes, in particular near-infrared fluorescent or dual-labeled near-infrared fluorescent and SPECT probes, or perhaps even MRI and CT. In this case, the large size of your delivery vehicle means that it should dominate the pharmacokinetics of the delivery process and are much less likely to change significantly whenever you put the fluorophore on or off. Indeed, a very smart method could be to hedge your bets, and this means to label those probes both with a SPECT probe and with a near-infrared probe and with perhaps a green probe, so that you can utilize all available different technologies in order to better evaluate this drug delivery vehicle, whether you're using microscopy, MR, optical imaging, SPECT imaging in small animals, or SPECT imaging in humans. Then you never have to worry about whether the pharmacokinetics of your probe is going to change, because it's always been the same no matter which imaging system and imaging modality you're using. Today I want to cover a couple of examples that highlight how you can use optical imaging and a little bit of how you can utilize gamma scintigraphy with SPECT probes to evaluate potential new drugs and drug delivery vehicles. One of these examples is getting drugs across the blood-brain barrier or into other blocked compartments in the body or protected compartments in the body is a particular challenge that's been out there for a long time. And indeed, a lot of work goes into developing ways to non-invasively transit the blood-brain barrier without actively disrupting it and destroying it, because the blood-brain barrier does a lot of important work for us. It keeps chemicals out of our brain that we need to keep out, and it keeps the ones in we need to keep in. People have also been interested in evaluating the targeting potential of large molecules like nanoparticles or peptides to deliver new therapeutics or existing therapeutics to tumor sites or to other sites of disease. In this case, people want to leverage the existing known biology of a therapeutic, but just get it to the target more effectively, more efficiently, and with more specificity. And optical imaging allows you a really rapid way to evaluate whether you're there or not. Finally, there's an interesting small class of potential drugs that are out there that are inherently near-infrared fluorescent. So why not take advantage of that property to track where the molecule is going in vivo and where it's concentrating, because, hey, it's already fluorescent. You don't have to label it with anything. So then your pharmacokinetics certainly don't change, and you don't have to use any additional chemistry in order to label it. So let's take a look at an example that was published a few years ago in neuroimaging that highlights some of the real utility of utilizing near-infrared fluorescence or tracking whether your delivery vehicle can make it into a targeted protected site or not. It had been reported that the peptide could effectively transit the blood-brain barrier and really go anywhere in the body with 100% efficiency. Now that turned out to be not entirely the case, so this group spent a decent amount of time trying to modify the peptide with other small molecules in order to see whether they could enhance the blood-brain barrier permeability of this particular peptide. And in this case, they IV injected it, imaged it in the near-infrared fluorescent region, and you can see a nice co-registration of what appears to be the brain with the brain of a mouse. Now, one of the really nice properties of using near-infrared fluorescence for imaging is that you can go between scales and instrumentation quite readily. So while the other image was able to nicely demonstrate that in fact they were getting nice targeting to what looks like the brain, it isn't clear exactly whether it's across the blood-brain barrier or not. However, because you don't have to change the label, the fluorophore, the near-infrared fluorophore is already on there, you can simply image those same brain slices that you take out of that brain using fluorescence microscopy. Now we can validate that in fact when you stain for endothelin, a marker of the blood vessels, that the near-infrared fluorescent labeled peptide, in this case labeled in red, had gotten not only into the brain but across the blood-brain barrier and was starting to illustrate its normal nuclear localization. Another group said, okay, that's pretty interesting, but another thing we'd like to be able to get across the blood-brain barrier, much more challenging than a peptide even, is a nanoparticle. And in this case, they utilized a very inexpensive commercially available fluorophore indocyanine green, and they loaded different amounts of, they loaded calcium phosphate nanoparticles with that ICG in order to track in vivo. Those calcium phosphate nanoparticles could then later be loaded with drug compounds. They then coated those calcium phosphate loaded nanoparticles with a variety of different large molecules, in this case gastrin and pentagastrin. And as you can see from the image, the in vivo image co-registered with X-ray, they were able to get what appears to be significant brain targeting, much more than with either the PEG-coated calcium phosphate nanoparticles alone in one, or the pentagastrin avidin-coated calcium phosphate nanoparticles in 3. Jean et al. Actually took another approach, and they said, well, what we'd like to do is take not just nanoparticles, but even larger particles like micelles that could be loaded with a variety of payloads quite readily, and see if we can deliver those across the blood-brain barrier. So they coated those nanoparticles with a peptide, KC2S, and they compared it to the appropriate negative control, in this case a near-infrared labeled PEG micelle. And they can see, in fact, that right away, even at 2 hours, they're starting to get more uptake of their micelles into the brain compartment than with the controls. And by 12 and 24 hours, there's a significant increase in the, really striking increase in the amount of micelle that's available in the brain compartment. Now, in both of these previous cases, it would be nice to see some additional work to in fact confirm that in various other mouse models, they were able to not only get it to be more effectively delivered to the brain, but across the blood-brain barrier in order to deliver some interesting payloads. This same group had another interesting challenge, which is to target micelles specifically to glioblastomas rather than the brain. In this case, they took a different peptide that's non-toxin, CDX, and they labeled their micelles with infrared fluorophore, coated it with their target peptide, and then injected it into glioblastoma-bearing mice. On the right-hand side of each panel in Figure A, you see a micelle loaded with near-infrared fluorophore that has CDX peptide on the surface. And right away, you can see, even without further quantification, that there's a significant increase in the amount of fluorophore that's reached the brain compartment at all time points. And really by 2 to 4 days, there's a marked improvement. They can then take this out ex vivo and show that in fact it's getting to the glioblastoma area. The interesting thing about this challenge is that the glioblastomas themselves, in some cases, disrupt the blood-brain barrier, making this problem a little bit easier. And it's the first of several examples I want to show where people are trying to utilize near-infrared fluorescence to see how well they can target large payloads to tumors. And there is, I would say, intermediate success with this type of work. One of the biggest challenges that you'll see will come later in the talk whenever I illustrate that in really in nice solid tumors, it can be a challenge to get even perfusion of these large nanoparticles in micelles through the tumor. What's nice is that having an in vivo optical imaging system allows you to be able to reach that conclusion really quickly and quite decisively. Really, one of the other nice things about this particular study is that not only can we see increased uptake of the micelles being delivered to the glioblastoma, whenever they actually load these micelles with the drug, they actually improve survival significantly, and specifically for the CDX coating, which mirrors exactly what they saw from their in vivo optical imaging, which means that in this case you could utilize the in vivo optical imaging to predict the delivery and successful delivery of your therapeutic to the target site. This is a really nice example where I want to show the use of a nanoparticle and a multimodal nanoparticle that is targeted and carries a fluorescent payload in order to image it, and an siRNA payload to mimic siRNA drug delivery, which is quite another interesting area of I would say, modern drug delivery, which is to not only look at delivering classic small molecules in order to treat diseases and kill tumors, but deliver larger payloads like siRNAs to silence specific genes or sets of genes in order to effectively treat the disease or kill the tumor. In this case, the group started with a magnetic nanoparticle, coated it with Cy5.5 for imaging, MPAP as their targeting agent, and siRNA for GFP as their mock drug delivery payload. They then placed two tumors on the flank of a mouse. In this case, one tumor that expresses RFP and one tumor that expresses GFP. And you can see from the whole body in vivo imaging that of the near-infrared fluorescent signal, that they're getting effective targeting and accumulation of their nanoparticles to both of those tumors. What's really interesting about having this siRNA for GFP as your evaluation is it allows you to non-invasively and both in vivo and in vitro demonstrate that the siRNA specifically silenced the GFP positive cells. So not only was it being delivered, as we could see from the near-infrared fluorescent channel, but it was also effective, as we can see from the GFP channels, both in vivo and in vitro. So you can, of course, always quantify that data as well and show that there's, by RT-PCR, for example, as an independent method to validate your in vivo imaging data. I want to point out this figure below. And this figure is important because it highlights the different distribution ex vivo, where we take the tumor out, slice it, put it back on the imaging system, and see different levels of penetration of the nanoparticles to different parts of the tumor, even in the same mouse in the same body. Again, the nice thing about fluorescence is we can leverage those fluorophores to image at multiple different scales to get it more and more information. So we can optimize the delivery in vivo using our whole animal imaging system. We can check it ex vivo using the zoom capabilities of the whole animal imaging system, and then confirm the subcellular and cellular distribution by fluorescence microscopy, all on the same animals, all with the same injection of probes, so they're all directly correlated to each other. And you don't have to go out and get a new secondary antibody to probe your targets in your microscopy examples. So this is an important, this article highlights what I believe is an important thing to keep in mind when developing large drug vehicles that have to target solid tumors, which is that even with the same tumors in the same mouse that see this roughly the same concentration of drug delivery vehicle, you get differential permeability into these tumors. Another group was interested in looking at a peptide that could be specifically targeted to the PSMA receptor. In this case, you can see that the peptide was labeled with a chelator, and that chelator was then filled with a SPECT imaging agent, Tec-99m. That SPECT chelator can be imaged not only in a 3D SPECT system like the Albira, but can also be screened in a very high throughput manner on the radioisotopic screen of the in vivo MSFX Pro and in vivo Extreme, which is a unique feature to in vivo preclinical molecular imaging systems. What's nice is that here you can evaluate the targeting specificity of your peptide rapidly using the imaging agent by looking at a cold block experiment, by looking at, as well as looking at important genetic negative controls, such as tumors that don't have the receptor. Because they could tell from the in vivo imaging experiment that this particular peptide could tolerate the additional chemical functionality of a chelator, they thought maybe they could add on to that a toxin and turn this into a targeted drug delivery agent. And in fact, they were able to do that. The cold block experiment worked just as well in the survival curve as it worked in blocking the uptake of the radiolabeled peptide. So that you can see here by looking at the mean tumor volume. So without treating with the drug with saline, for example, the tumor continues to grow just fine. If you add in your targeted peptide, your peptide that targets the tumor cells specifically, you see significant reduction really as early as day 4 in the size of the tumors. And whenever I put in a competitive blocking agent that keeps my peptide from reaching the tumor, now my tumors grow just fine. So that mimics beautifully what we saw in the in vivo in vivo molecular imaging experiment, which is that you were getting specificity of this peptide to the tumors, and it could deliver a large payload, relatively large payload, such as a chelator with a metal, or in this case, a drug. Finally, next I want to give an example of looking at small molecule antibodies rather than small molecules. People have been interested in utilizing antibodies as a therapeutic for quite a long time. Antibodies, or probably more importantly antibody mimetics, are able to deliver peptides and other payloads, including drugs or micelles carrying drugs, specifically to target sites. Hopefully tumors, could be a lot of different other applications, including with cardiovascular disease. In this case, they just wanted to see how well their antibody was being targeted to a tumor, or whether it was in fact being targeted to some other part of the body that they don't want it to be targeted to. There's a lot of toxicity in vivo due to liver toxicity, kidney toxicity, and cardiac toxicity that can really kill a drug in clinical trials. And we want to identify those antibodies that bind to those sites right away and get rid of them. So in this case, they took their antibody and labeled it with Alexa 680, which is found in commercially available fluorescent labeling kits. And unfortunately, they could clearly see in a very simple experiment that the antibody was targeting the kidneys and not targeting the tumors. So they can just throw that antibody away. Nice idea, but definitely don't let that proceed through the drug discovery process. Better luck with this particular antibody, where again it was labeled with Alexa 680, image just like before, 24 hours post-injection, and you can see very nice targeting to their subcutaneous tumor implants, and no targeting to the kidneys. So good news there. And what's great with these whole animal in vivo imaging systems is that you don't have to image just one mouse at a time. With the Extreme and the MSFX Pro, you have a 20-centimeter platen, so you can easily image 5 mice or more at a time in a single acquisition to really drive your throughput of these experiments. You can also utilize probes for more than just oncology, although we've looked at a lot of oncology models. In this case, a group was looking at loading micelles with DIR as well as anti-inflammatories and using those to see if they can improve delivery of known anti-inflammatories to an arthritic lesion. And you can see by looking in the near-infrared channel after you've labeled these micelles and injected them IV that you get very nice targeting to the site of inflammation. Now this image also tells you something else, thinking back to our previous image. Most of these micelles are being taken up and cleared through the liver. That tells us that in our drug discovery process we need to pay careful attention to whether there's any liver toxicity, or if what we're loading into these micelles is known to be liver toxic already, it's probably not a good idea to carry it forward. In this case, they didn't see that. And in fact, importantly, they were able to, with the treatment of the liposomes or micelles, reduce the clinical severity of the arthritis in these models. My last example is to illustrate how we can take advantage of drugs or drug-like molecules that themselves may be near-infrared fluorescent. In this case, we're looking at a class of compounds known as porphyrazines that not only could act as fluorescent probes, but also could act as photodynamic therapy agents. Indeed, many photodynamic therapy agents will have near-infrared fluorescent properties, or at the very minimum fluorescent properties that you can take advantage of. The particularly interesting thing about this scaffold is that it emits not only in the near-infrared, but also in the visible. So again, I can check my distribution through whole animal imaging and switch to microscopy and work back in the visible in a lot of standard microscopes that are out there everywhere, all without switching the probe. This group also took advantage of the idea that they wanted to make their tumors labeled with RFP so that they could co-register whether their near-infrared fluorescent porphyrazines were not only getting throughout the body, but whether they were co-localizing with those RFP-positive tumors. And you can see the individual channels in A and B co-registered with X-ray, and then really the overlapping signal can be quite easily seen in yellow here in panel C. You could also confirm that by microscopy again, that the green, in this case E, remember this porphyrazines emit not only in the near-infrared but also in the green, was taken up by the cells not only at the surface but also actually internalized into the cells and co-registered quite nicely with the RFP-positive tumor cells. So in conclusion, I highlighted a couple of areas where molecular imaging, in particular near-infrared fluorescent optical imaging and nuclear imaging, can really aid in the rapid evaluation of novel vehicles for delivering drugs to protected sites like across the blood-brain barrier, into tumors, and being able to rapidly identify the challenges that you might have in delivering your probes to tumors, whether it be off-target kidney binding or non-ideal, non-even perfusion of your solid tumor with the nanoparticle, as well as the ability to actually track the small subset of molecules that are near-infrared fluorescent, particularly whenever they concentrate at your target site. This allows you to really rapidly, up to 5 mice at a time, with just a simple 5 to 30-second imaging experiment, get quickly to the answer that you really need to know. Do these new vehicles get to the site? Is it specific? And does more of it get there than to potentially potential off-target organs? So I'd like to thank you all for your attention, and I'd be glad to take any questions that you might have.